The next family of organisms we're going to talk about are the Chlamydiaceae. Lots of high-yield organisms here. We're going to talk about Chlamydia trachomatis, very highly testable on step 1 of the USMLE. We're also going to talk about Chlamydiophila pneumoniae and Chlamydiophila psittaci. So we're going to talk about all 3 of those, but before we get into specific diseases, let me introduce some of the features of this family. This is the first of the bacteria that we're going to start talking about that is actually an obligate intracellular bacteria. Most of the bacteria we've talked about thus far are facultative intracellular, so they get into a macrophage and then survive killing by the macrophage. In this case, chlamydia cannot replicate outside of a host cell. Okay? So, why? The main reason is because it cannot make its own ATP. So it's utilizing the host cell to make ATP. One of the unique features of the family is in the second point that I'm circling for you here, the elementary body versus the reticulate body. This is unique really to the Chlamydiaceae family, where you need actually 2 forms of the organism in order to propagate yourself. Now, the elementary body, and this is testable. I'll give you an example of when this is testable, so stick with me here. The elementary body is the infectious form of the bacteria. So you have an elementary body attaching to the host cell. The reason the elementary body is needed for that is the infectious form is because it has the attachment proteins required to attach to the host cell, all right? So elementary body is the infectious form. Now, once that elementary body gains access to the host cell, it will convert to the reticulate body. If you can remember R, reticulate, replication, reticulate, replication. The reticulate body is the replicative form of chlamydia. So now you have the elementary body, it gains access into the cell, converts to a reticulate body. The reticulate body replicates in the cell. Now here's an important point. Before those organisms can leave the cell and go on to infect another cell, what are they going to have to do? They're going to have to convert back to an elementary body, or they will not have the proper receptors to gain access to a host cell. How on earth are they going to ask you this? Well, very simply. They can give you a nice little vignette about non-gonococcal urethritis, and they can say, which of the following structures are used for this organism to gain access to host cells? Or which of the following is used for replication of the organisms in the above scenario? So then you'll have to know elementary body and or reticulate body. So that's how they can easily turn that basic information into a clinically relevant scenario. So you don't want to forget about that. So again, we're going to focus predominantly on Chlamydia trachomatis. It's the largest group here, causing a lot of different diseases actually, and those diseases are differentiated based on the serotype. Then we will focus on the Chlamydiophila pneumoniae and psittaci. So again, features here, we can't make ATP, we're obligate intracellular, and we cannot be seen on the gram stain. So I'm going to help you out with some diagnostic tools to help you figure out whether or not you may have a chlamydia infection. What's the reservoir for Chlamydia trachomatis? It's typically a sexually transmitted disease, but can also cause eye infections as well in areas where certain serotypes of Chlamydia trachomatis are very prevalent. So as far as transmission, again, sexual contact, but as far as trachoma, which is the eye infection caused by serotypes A through C of Chlamydia trachomatis, it's from contact, hand-to-eye contact, or sometimes flies. Flies that might land on someone's eye, pick up an organism, and go land on someone else, transmitting that organism. Now, the pathogenesis will destroy some of the epithelial cells, causing damage, and you're going to get a granulomatous type response. Very similar to what we've talked about with a lot of those facultative intracellular organisms. But you will get a lot of inflammation. So the first specific disease we're going to talk about for Chlamydia trachomatis are the STDs. This is the most common bacterial STD in the U.S., okay? Most common bacterial STD in the U.S. So you're given a vignette, and you've got a 28-year-old male that presents to his PCP with burning upon urination, maybe a little bit of a fever. They don't tell you anything about gram stain. They might mention that he did have a urethral exudate, and they just want to know what's most likely. Chlamydia is most likely. It is more common than gonorrhea in the U.S. Now, it's not the most common STD in the U.S. Herpes and HPV are really duking it out for first place there, but as far as bacterial infections, it's the most common. Now, unfortunately here, in addition to knowing the organism, you do have to know the serotypes. So I'm going to talk about the serotypes for sexually transmitted diseases are D through K. Now, I know you're very clever. You come up with mnemonics all the time. I'm sure that you can think of a mnemonic to help remind you the serotypes of Chlamydia trachomatis that are most prevalent in the U.S., D through K. You're going to basically fill in with another couple letters, all right? What does it cause? Non-gonococcal urethritis. We have a lot of infertility in the U.S. and pelvic inflammatory disease that goes back to the fact that chlamydia infections are very, very common in the U.S. What other diseases you can see with the same serotypes? If you have a mother who is positive for chlamydia, she can actually pass that on to her child, and they can get inclusion conjunctivitis and or pneumonia in an infant. All right, another type of disease caused by the same organism but different serotypes is something called lymphogranuloma venereum, or LGV. Now, the serotypes here are very easy to remember because they all start with L, so you're not going to have any trouble remembering the serotypes responsible for LGV. This is not a very common disease in the U.S. It's prevalent in Africa and Asia and South America as well, but again, may at least show up on a distractor list for an STD, so you definitely want to be aware of it. The disease LGV is interesting because it is not the classic cause of what we consider for elephantiasis, but it works in much the same way. So what happens is you get swollen lymph nodes in the genital area, and it starts to block the flow of lymphatics. So you get massive lymphedema, much like you would with elephantiasis from the parasite, okay? So you get swollen lymph nodes and lymphedema due to the fact that you're blocking the flow of the lymphatics. Now, what can happen is those lymph nodes can be so swollen, they'll start to the skin over top of them will get really, really thin. It'll start to suppurate and ooze from that. And in addition, you can also see fistulas as well from LGV. Again, not very common in the U.S., so you got to keep that in mind on the United States medical licensing exam. Last, leading up the rear here, bringing up the rear, is trachoma. Trachoma is due to serotypes A through C. Okay, so we have non-gonococcal urethritis, D through K, LGV, all the serotypes that start with L, and then trachoma is A through C. This is not common in the U.S., but in areas where this is very prevalent, in Africa, for example, it is the leading cause of preventable blindness. And it's not that you get this infection and you go blind immediately, but what happens is you come in contact with trachoma, again, remember, from contact with the eyes or a fly going from one person's eye to another, and you get a lot of inflammation. So what happens is your eyelashes, you get a lot of scar tissue built up and inflammation, your eyelashes start to turn inward and over time begin to abrade the cornea. And that going on for a long period of time causes scarring and will eventually lead to blindness. So this is usually the result of repeated infections in endemic areas. How are we going to diagnose chlamydia? Again, they're not probably going to tell you, we did a DNA probe for chlamydia and it was positive. No. What they're going to do is you're going to have to base it on the clinical presentation and what's most likely. Here's an example of what you might see for chlamydia. If we're talking about non-gonococcal urethritis, you get a presentation of a male. They're coming in with this urethral exudate, and you gram stain that exudate, and what are you going to see? Are you going to see organisms? You're not going to see organisms because remember, chlamydia doesn't gram stain. Are you going to see neutrophils or PMNs? Absolutely. You're going to see PMNs because there's a lot of inflammation. You've got urethritis. So, if they mention we gram stained the urethral exudate, we found PMNs, but we couldn't detect any organisms, a light bulb should start to go off in your head. Ding, 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 ding. Wow, this is something. We know we have inflammation. We got neutrophils. What could it be? And chlamydia is something that would definitely be a positive answer, okay? We can't really culture this per se in the lab. Why? Because it's obligate intracellular, so keep that in mind. Now, how are we going to treat it? We treat it with doxycycline or erythromycin. We can prevent disease when we're talking about transmission of disease from mother to infant. We can use systemic erythromycin, for example, to help prevent pneumonia. But as far as real prevention by way of vaccine, we don't have one. Very difficult to control the spread of chlamydia.